Plant tissue culture. MCQs. Which of the following is not an application of plant tissue culture? A. Synthetic seed production. B. Germplasm conservation. C. Elimination of pathogens. D. Production of animal insulin. Answer: D. Production of animal insulin. Tissue culture is used only in plants, not for producing animal-derived hormones. Which technique is particularly useful for conserving rare and endangered plant species? A. Organ culture. B. Callus culture. C. Cryopreservation. D. Anther culture. Answer: C. Cryopreservation. Germplasm of rare species is preserved at ultra-low temperatures using cryopreservation. Which of the following is a limitation of clonal propagation? A. High rate of somaclonal variation. B. Limited genetic diversity. C. Very slow multiplication. D. No commercial use. Answer: B. Limited genetic diversity. Since all plants are clones, there is little genetic variation, making them vulnerable to diseases. What is the major advantage of synthetic seed technology, via tissue culture? A. Easy transport and storage of elite genotypes. B. Increased genetic variation. C. Eliminates the need for culture medium. D. Avoids need for sterilization. Answer: A. Easy transport and storage of elite genotypes. Synthetic seeds allow encapsulated somatic embryos to be stored and transported like true seeds. Which of the following is a pharmaceutical product obtained through plant tissue culture? A. Penicillin. B. Artemisinin. C. Streptomycin. D. Tetracycline. Answer: B. Artemisinin. Artemisinin, antimalarial drug, is produced from artemisia using tissue culture. An advantage of tissue culture over conventional breeding is: A. Requires no sterile conditions. B. Produces genetically variable plants. C. Faster and large-scale multiplication. D. Can be performed by unskilled labor. Answer: C. Faster and large-scale multiplication. Tissue culture is much faster and produces thousands of clones compared to conventional breeding. Which of the following is a disadvantage of plant tissue culture? A. Requires aseptic conditions. B. Somaclonal variation may occur. C. Expensive infrastructure. D. All of the above. Answer: D. All of the above. All these are limitations of plant tissue culture. Which of the following explains why tissue culture plants are often more expensive than conventionally grown plants? A. Requirement of growth regulators and nutrients. B. Need for sterile environment and skilled labor. C. Use of controlled growth chambers. D. All of the above. Answer: D. All of the above. Tissue culture demands strict sterile conditions, expensive chemicals, and high-skilled labor. Which type of culture can directly produce haploid plants? A. Meristem culture. B. Anther culture. C. Organ culture. D. Suspension culture. Answer: B. Anther culture. Anther, or pollen, culture can produce haploid plants, useful in crop breeding. Which of the following is a limitation of somaclonal variation? A. Produces genetically identical plants. B. May produce undesirable mutations. C. Always produces disease-free plants. D. Eliminates the need for micropropagation. Answer: B. May produce undesirable mutations. Somaclonal variation sometimes gives useful traits, but often results in undesirable mutations. Which of the following crops is most successfully micropropagated for large-scale commercial farming? A. Rice. B. Banana. C. Maize. D. Wheat. Answer: B. Banana. Banana is one of the most common commercially micropropagated crops, providing disease-free planting material. Which product obtained through plant tissue culture is widely used as an anti-cancer drug? A. Taxol. B. Insulin. C. Morphine. D. Penicillin. Answer: A. Taxol. Taxol, from Taxus cell cultures, is used in chemotherapy for cancer treatment. Which plant tissue culture-derived compound is an antimalarial drug? A. Reserpine. B. Artemisinin. C. Vincristine. D. Agmelecin. Answer: B. Artemisinin. Artemisinin, obtained from Artemisia annua, is an effective antimalarial drug. Vanillin, a flavoring agent in chocolates and ice creams, can be obtained through: A. Suspension culture of Vanilla planifolia. B. Root culture of Solanum tuberosum. C. Callus culture of Catharanthus roseus. D. Embryo culture of ZMAs. Answer: A. Suspension culture of Vanilla planifolia. Tissue culture of vanilla is used for sustainable production of vanillin flavoring. Which compound from Catharanthus roseus tissue culture is used as an anti-cancer drug? A. Taxol. B. Reserpine. C. Vincristine. D. Morphine. Answer, C. Vincristine. Vincristine and vinblastine are alkaloids derived from Catharanthus rosea cell cultures and used in cancer therapy. Which of the following flavors/additives is produced using plant tissue culture? A. Aspartame. B. Menthol. C. Citric acid. D. Caffeine. Answer, B. Menthol. Menthol, used in toothpaste, chewing gum, and candies, can be produced via tissue culture. Chiconin, obtained from plant tissue culture, is mainly used in A. Anti-malarial drugs. B. Wound healing, cosmetics, and natural dyes. C. Anti-hypertensive medicines. D. Fertilizer production. Answer, B. Wound healing, cosmetics, and natural dyes. Chiconin, a red pigment from lithospermum cultures, is used in cosmetics and medicine. Which tissue culture-derived compound is used in hypertension treatment? A. Agmelecin. B. Taxol. C. Chiconin. D. Vanillin. Answer, A. Agmelecin. Agmelecin and reserpine, from Rauwolfia serpentina, are anti-hypertensive drugs obtained via culture. Which of the following is a direct product of tissue culture widely used in daily dietary crops? A. Clonal bananas and potatoes. B. Edible vaccines. C. Synthetic seeds only. D. Hybrid maize. Answer, A. Clonal bananas and potatoes. Banana and potato are major crops mass-produced using tissue culture for daily consumption. Capsaicin, obtained via chili, capsicum, cultures, is used in A. Coloring textiles. B. Food flavoring and pain relief creams. C. Fertilizer production. D. Baking powder. Answer, B. Food flavoring and pain relief creams. Capsaicin adds spiciness to foods and is also used in topical ointments for pain relief. Which of the following ornamentals is extensively propagated by tissue culture? A. Lotus. B. Orchids. C. Mango. D. Rice. Answer, B. Orchids. Orchids are among the top ornamental plants produced commercially via tissue culture. Which timber/plantation crop is commonly propagated by tissue culture for industrial use? A. Wheat. B. Teak. C. Maize. D. Rice. Answer, B. Teak. Teak, eucalyptus, bamboo, and sandalwood are propagated by tissue culture for timber and paper industries. Which of the following is an example of biopharmaceutical produced in plants using tissue culture plus genetic engineering? A. Plantibodies, plant-made antibodies. B. Insulin. C. Streptomycin. D. Tetracycline. Answer, A. Plantibodies, plant-made antibodies. Plantibodies are therapeutic antibodies produced in plants through tissue culture combined with genetic engineering. Edible vaccines are being developed in which tissue culture-based crops? A. Potato and banana. B. Rice and wheat. C. Maize and tomato. D. Carrot and spinach. Answer, A. Potato and banana. Potatoes and bananas are engineered via tissue culture to produce edible vaccines for diseases like hepatitis B. Which of the following is a daily life product from tissue culture used in perfumes and flavor industry? A. Artemisinin. B. Menthol. C. Vinblastine. D. Reserpine. Answer, B. Menthol. Menthol is widely used in perfumes, flavors, and medicines. Which plant tissue culture product is widely used as a food preservative and additive? A. Vanillin. B. Reserpine. C. Chiconin. D. Taxol. Answer, A. Vanillin. Vanillin is a flavoring and food additive obtained from vanilla cultures. Which plant tissue culture derived compound is used in the treatment of hypertension? A. Vinblastine. B. Agmelecin. C. Chiconin. D. Taxol. Answer, B. Agmelecin. Agmelecin and reserpine are anti-hypertensive alkaloids derived from Rauwolfia serpentina. Which of the following best represents daily life uses of tissue culture plants? A. Banana, potato, orchids, menthol, vanillin. B. Penicillin, insulin, cheese, coffee. C. Wheat, rice, tea, cotton. D. Bacteria, fungi, yeast, antibiotics. Answer, A. Banana, potato, orchids, menthol, vanillin. These are common food, ornamental, and flavor products from plant tissue culture. Which product of tissue culture is being explored as a natural food coloring? A. Chiconin. B. Taxol. C. Artemisinin. D. Capsaicin. Answer, A. Chiconin. Chiconin is a natural red dye used in cosmetics and food coloring. Which of the following combinations is correctly matched? A. Taxol rightward arrow anti-malarial drug. B. Vanillin rightward arrow perfume and flavor industry. C. Capsaicin rightward arrow fertilizer. D. Reserpine rightward arrow food additive. Answer: B. Vanillin rightward arrow perfume and flavor industry. Vanillin is used in perfumes and as a flavoring agent, while Taxol is anti-cancer, capsaicin is a spice/pain reliever, and reserpine is anti-hypertensive. Who first demonstrated the concept of plant tissue culture by regenerating a whole plant from carrot cells? A. Haberlandt. B. Skoog and Miller. C. Stewart. D. Murashige and Skoog. Answer: C. Stewart. F.C. Stewart, 1958, regenerated carrot plants from cultured cells, proving totipotency. The totipotency of plant cells refers to A. Ability to photosynthesize. B. Ability to produce callus only. C. Ability to develop into a complete plant. D. Ability to absorb nutrients. Answer: C. Ability to develop into a complete plant. Totipotency is the capacity of a single cell to regenerate into a full plant. The MS medium was developed in 1962 by A. White and Haberlandt. B. Murashige and Skoog. C. Stewart and Miller. D. Knopp and Gamborg. Answer: B. Murashige and Skoog. Murashige and Skoog formulated MS medium in 1962, the most widely used culture medium. Which of the following pairs of growth regulators is essential in most tissue culture experiments? A. Auxins and cytokinins. B. Ethylene and ABA. C. Gibberellins and ethylene. D. Cytokinins and ABA. Answer: A. Auxins and cytokinins. Auxins and cytokinins regulate callus induction, shoot, and root differentiation. Which plant part is commonly used for embryo rescue culture? A. Endosperm. B. Immature embryo. C. Root tip. D. Meristem. Answer: B. Immature embryo. Embryo rescue helps in hybridization where immature embryos may not survive naturally. Which of the following is an example of secondary metabolite production in suspension cultures? A. Vanillin. B. Artemisinin. C. Taxol. D. All of the above. Answer: D. All of the above. Explanation: suspension cultures are used to produce many secondary metabolites like vanillin, artemisinin, and taxol. Which factor most significantly affects somaclonal variation? A. High concentration of growth regulators. B. Use of solid medium. C. Use of sucrose as carbon source. D. Low light intensity. Answer: A. High concentration of growth regulators. Excessive use of auxins and cytokinins increases genetic instability, causing somaclonal variation. Synthetic seeds are produced using A. Anther culture. B. Meristem culture. C. Encapsulation of somatic embryos. D. Suspension culture. Answer: C. Encapsulation of somatic embryos. Somatic embryos are encapsulated in a gel-like material to form synthetic seeds. Which of the following techniques helps in haploid plant production? A. Embryo rescue. B. Meristem culture. C. Anther or pollen culture. D. Organ culture. Answer: C. Anther or pollen culture. Anther/pollen culture leads to haploid plant production, useful in plant breeding. Which compound is used as a carbon source in most plant tissue culture media? A. Glucose. B. Maltose. C. Sucrose. D. Fructose. Answer: C. Sucrose. Sucrose is the most commonly used carbon source in plant tissue culture. Which step is performed at the very end of plant tissue culture before field transfer? A. Callus induction. B. Acclimatization, hardening. C. Organogenesis. D. Sterilization. Answer: B. Acclimatization, hardening. Hardening prepares plantlets for field conditions by gradually exposing them to natural environment. Which is a limitation of plant tissue culture in terms of biodiversity? A. Increases variability. B. Reduces genetic diversity due to clonal propagation. C. Always produces hybrids. D. Prevents cross-pollination. Answer: B. Reduces genetic diversity due to clonal propagation. Since plants are identical clones, biodiversity decreases, making them vulnerable to diseases. Which of the following statements about cryopreservation is correct? A. Used for short-term storage of germplasm. B. Storage at -196 degrees Celsius using liquid nitrogen. C. Limited to ornamental plants only. D. Cannot conserve endangered species. Answer: B. Storage at -196 degrees Celsius using liquid nitrogen, Cryopreservation conserves germplasm long-term at -196 degrees Celsius. The production of edible vaccines in plants is an example of A. Protoplast fusion. B. Transgenic technology plus tissue culture. C. Somaclonal variation. D. Callus culture only. Answer: B. Transgenic technology plus tissue culture. Genetic engineering plus tissue culture is used to develop edible vaccines in crops like potato and banana. Which plant-derived drug obtained from tissue culture is used in cardiac treatment? A. Digitalis. B. Artemisinin. C. Taxol. D. Chicanin. Answer: A. Digitalis. Digitalis, derived from Digitalis purpurea, is used to treat heart conditions. Which of the following is an advantage of in vitro conservation, tissue culture, over field conservation? A. Requires less land and resources. B. Faster multiplication of rare plants. C. Long-term storage without environmental risk. D. All of the above. Answer: D. All of the above. In vitro conservation is efficient, space-saving, and safer compared to field conservation. Which of the following growth regulators is not commonly used in plant tissue culture? A. Auxins. B. Cytokinins. C. Abscisic acid. D. Ethylene. Answer: D. Ethylene. Ethylene is generally inhibitory to growth and not preferred in tissue culture. The callus is best described as A. A cluster of differentiated root cells. B. An unorganized mass of undifferentiated cells. C. A suspension of protoplasts. D. A collection of haploid cells. Answer: B. An unorganized mass of undifferentiated cells. Callus is a mass of undifferentiated parenchyma cells formed from an explant. Which is a key advantage of plant tissue culture in pharmaceutical production? A. Unlimited, controlled production of valuable secondary metabolites. B. Elimination of all genetic variation. C. Easier than chemical synthesis of all drugs. D. Can only be used for food crops. Answer: A. Unlimited, controlled production of valuable secondary metabolites. Tissue culture enables stable and large-scale production of alkaloids, pigments, and drugs. Which of the following best summarizes the importance of plant tissue culture in daily life? A. Provides only ornamental plants. B. Limited to genetic studies. C. Supplies food crops, medicines, flavors, dyes, and conservation tools. D. Only used in laboratories with no practical use. Answer: C. Supplies food crops, medicines, flavors, dyes, and conservation tools. Tissue culture plays a vital role in agriculture, pharmaceuticals, food industry, and biodiversity conservation.